The Monomethyl Auristatin E is a synthetic antimitotic agent derived from the natural product 𝗕𝗟𝗖𝗦𝗘𝗗 10. This highly potent cytotoxin has gained prominence as a payload in antibody-drug conjugates ADCs. Its mechanism of action involves the inhibition of tubulin polymerization, leading to cell cycle arrest and apoptosis. Notably, MMAE features a cleavable linker compatible with various tumor-specific targeting moieties. This flexibility has facilitated its widespread use across numerous ADC clinical candidates. In this report, we will discuss the top FDA or EMA-approved MMAE-based ADCs as well as those in active investigation with an emphasis on their linker technology. We will also analyze their commercial performance and projected growth as well as conduct an in-depth examination of the intellectual property IP environment to identify key patents and highlight considerations for due diligence. The MMAE is a microtubule inhibitor that induces mitotic arrest by binding to tubulin dimers, thereby preventing their polymerization into microtubules. This disruption of microtubule dynamics affects crucial cellular processes such as chromosome segregation, spindle formation, and cell cycle progression, ultimately leading to apoptosis. The molecule's structure enables site-specific conjugation of various antibody-drug pairs. Moreover, the linker employed in these conjugates plays a significant role in determining the pharmacokinetic profile and therapeutic efficacy of the final ADC. We present the top 3 FDA or EMA-approved MMAE-based ADCs listed below. The table provides a concise overview of each drug, including target, indications, approval year, linker chemistry, and drug-antibody ratio (DAR). Additionally, we include sales data for these drugs. For each, we have compiled 5 years of historical sales figures along with projections for the next 3 years. Furthermore, we provide insights into the IP landscape associated with each drug, specifying who holds the patents for the antibody, linker, payload, and conjugation methods. Information on patent expiry dates is also included. The first drug in our list is brentuximab vedotin, marketed under the brand name Adcetris. It is indicated for the treatment of Hodgkin lymphoma and systemic anaplastic large cell lymphoma. The drug was initially approved by the FDA in 2011 and by the EMA in 2012. Brentuximab vedotin features a protease cleavable valine-citrulline valsate linker. The DAR of the drug is approximately 4 since its launch. Adcetris has achieved significant sales milestones, reaching $1.2 billion in 2022. Projections indicate that sales will continue to grow steadily over the next 3 years, reaching an annual rate of $1.5 to $1.7 billion between 2025 and 2027. Seagen Inc. holds the patents for the antibody, linker, payload, and conjugation methods associated with brentuximab vedotin. These patents are set to expire between 2020 and 2031. Polatuzumab vedotin, marketed as Polivy, is indicated for the treatment of relapsed or refractory diffuse large B-cell lymphoma (DLBCL). Approved by the FDA in 2019 and the EMA in 2020, polatuzumab vedotin also utilizes a valsate cleavable linker and has a DAR of approximately 3.5. Polivy generated sales of $500 million in 2020, to which is expected to grow to $800 million to $1 billion annually by 2025 to 2027. Genentech, a Roche company, holds the patents for the antibody conjugation chemistry and linker composition of polatuzumab vedotin. These patents are set to expire between 2029 and 2030. Enfortumab vedotin, marketed under the brand name Padcev, is indicated for the treatment of metastatic urothelial carcinoma. Approved by the FDA in 2019, Padcev features a malamidocapro-MC valsate cleavable linker and has a DAR of approximately 3.8. Padcev has demonstrated steady growth in sales, reaching $400 million in 2022. This growth is projected to continue at an annual rate of $900 million to $1.2 billion by 2025 to 2027. The intellectual property for Padcev is jointly owned by Seagen and Astellas. They have filed multiple patents related to the linker technology and conjugation strategies. The patents held by Seagen are set to expire between 2029 and 2030, while those held by Astellas are set to expire between 2028 and 2031. 𝗕𝗟𝗢𝗦𝗧𝗘𝗥, marketed as Tivdak, is a promising ADC targeting tissue factor TF currently approved for metastatic cervical cancer by the FDA in 2021. However, EMA approval is still pending. Tivdak features a cleavable peptide linker and has a DAR of approximately 4. In its first full year of sales in 2022, Tivdak generated $100 million in revenue, which is projected to reach $300 to $500 million annually by 2025 to 2027. The patents for Tivdak are shared between Seagen and Genmab, with some owned by Seagen dating back to 2008 and others jointly owned with Genmab dating back to 2010. These patents cover various aspects of the linker, antibody, and the combination of both for use in cancer therapy. 𝗕𝗟𝗢𝗕𝗗𝗏 is an experimental ADC targeting Nectin for currently in phase 1 to clinical trials for advanced solid tumors. Developed by 9m Biopharma, this ADC uses a novel site-specific linker and has a homogeneous DAR. The estimated peak sales for 9 megawatts to 8 to 1 are projected to reach $500 million to $1 billion annually, with an anticipated approval by 2026. However, as this asset is still in early development, limited patent information is publicly available. In conclusion, MMAE-based ADCs represent a class of therapeutics with immense potential for treating various cancers. The commercial success of approved MMAE-based ADCs, such as Adcetris, Polivy, and Padcev, demonstrates the viability of this approach. Moreover, the promising performance of investigational candidates like 𝗕𝗟𝗢𝗦𝗧𝗘𝗥 and 9 megawatts to 8 to 1 suggests a bright future for this drug class. Notably, the next generation of MMAE-based ADCs is characterized by improved linker technology, homogeneous DARs achieved through site-specific conjugation methods, and robust IP protection. Careful consideration of key IP elements such as antibody patents, linker payload chemistry, conjugation techniques, and global coverage is crucial when evaluating these assets. A thorough due diligence process involving patent life, freedom to operate, and litigation risk assessments will ensure informed investment decisions. The continued development and optimization of MMAE-based ADCs hold immense promise for improving cancer treatment strategies. If you enjoyed this report, please hit the like button and subscribe for more updates. Thank you for watching.